In this video, we're going to expand upon our understanding of genetic equilibrium and look at one possible calculation that we can do involving populations that are in genetic equilibrium. Now, genetic equilibrium is the state of conditions where the allele frequencies, that being how likely it is to observe a specific phenotype within the population, remain constant over multiple generations. So as we can see here, the parent generation has only pink colored flowers, and this is also true in the offspring generation. And if this trend were to continue, this would mean that no observable evolution is taking place because there is no selection pressure that is favoring the selection of a different phenotype over the pink flowered phenotype here. Now, if the population is in genetic equilibrium, this means that it is technically possible to calculate what the frequencies of each specific phenotype would be within the given population, which brings us to one of the few situations where we can actually use math in the context of evolution, albeit a fairly simple equation. And this equation was developed by an English mathematician who rather famously devised this equation on a cocktail napkin in a pub. And his name was G.H. Hardy, which is why the equation that we're going to be looking at is what we call the Hardy-Weinberg genetic equilibrium. And we can actually trace the origin of this calculation by looking at the Punnett square. So this supposedly was done when G.H. Hardy had a conversation with Reginald Punnett, the English geneticist who devised the Punnett square, in order to predict allele frequencies if we knew what dominant and recessive alleles were. And what G.H. Hardy designed this equation to do is to predict the frequency of homozygous dominant, heterozygous, and homozygous recessive alleles, although rather than referring to them as homozygous big A, heterozygous, or homozygous little a, we are going to be referring to the dominant and recessive alleles instead as P and Q. This is simply a convention. We will simply refer to P as the same allele as big A and Q as the same allele as little a, because that's what G.H. Hardy devised. So if we do a monohybrid cross, meaning that both of the parents are heterozygous for P and Q, so this particular sperm, if this is our father, is going to have the P allele, and this particular sperm is going to have the Q allele, and the same goes for the eggs if this is the mother. So as we expect from a typical monohybrid cross, this zygote is going to be homozygous dominant, and this one here and this one here are going to be heterozygous, and this one here is going to be homozygous recessive. This is nothing that we haven't seen before. Now from this, G.H. Hardy developed a relatively intuitive equation based on the probabilities that we see here. So the total percent of all alleles is obviously 100% here, and if we look at the probability of producing a homozygous dominant genotype, which is our PP, well, P times P is what G.H. Hardy called P squared, because P times P is, well, P squared, and then the heterozygous genotype, what we call PQ. As we see here, we see that unlike the homozygous dominant genotype, which has a 1 in 4 probability, the PQ genotype has a 2 out of 4 probability of being produced, simply because of the Punnett square, and therefore we use the variable 2PQ to represent the heterozygous genotype, simply because there is a twice as likely probability of getting this particular genotype here. And finally, the homozygous recessive genotype, QQ, if the homozygous dominant genotype is referred to as P squared, the homozygous recessive genotype would be q squared, which also has a probability of 1 over 4. And since G.H. Hardy was only looking at 2 alleles, the probability of getting, or rather the total percentage of each allele in the population is 100%, because these are the only 2 alleles that exist. And therefore, Hardy-Weinberg was able to come up with the formula p squared plus 2pq is equal to q, or plus q squared is equal to 1. That being 100% of all alleles within the population are determined by what percentage of your population is homozygous dominant, heterozygous, or homozygous recessive. Now, before we get to the actual calculations, it's important to consider that this formula can only be used under the conditions of genetic equilibrium, in which the allele frequencies are constant, because if natural selection were occurring, that would mean that one of the particular phenotypes displayed within the population is being favored over the other 2, which would mean that this calculation would be impossible to do, because these allele frequencies would actually be changing and not be constant, which would mean this mathematical equation is not true. So if we review the conditions of genetic equilibrium, this means that there is no natural favoring of one phenotype over another, meaning that the likelihood of each genotype surviving is going to be equal, meaning that there is effectively no natural selection going on within the environment, and natural selection, of course, is therefore caused by some sort of selection pressure, meaning that there is no significant selection pressure that is going to influence these allele frequencies in any way. We also need to restrict migration, or gene flow, if we prefer the genetics-related term, meaning that specific organisms with specific genotypes do not enter or leave the population, at least significantly enough to affect the allele frequency. Random mating occurs, meaning that sexual selection does not favor a specific phenotype, meaning that each specific organism with a specific genotype is equally likely to reproduce. And of course, we need to also consider that there is no significant mutation rate occurring within the population that can lead to a brand new allele being produced. So now that that's all out of the way, we can try a relatively simple practice problem here. So remember that for all intents and purposes, variable P is considered the dominant allele, so what we would use big A for, and variable Q is what little a is used for. So if we want to calculate what the frequency of the Q allele is going to be, remember that we actually have 2 equations, since P and Q are the only different alleles that occur within this specific gene. In this case, P plus Q is equal to 1. Therefore, since P is equal to 90%, all we need to do is subtract 90% from 100%, and therefore allele Q, the recessive allele, occurs 10% of the time, whereas the dominant allele occurs 90% of the time. Now, what this allows us to do is, if we actually put P and Q into the Hardy-Weinberg equilibrium equation, P squared plus 2pq plus q squared is equal to 1, we can actually substitute P in for P squared. So this would be 0.9 squared, and this would give us the allele frequency of the homozygous dominant allele here. Then if we put P and Q in for 2pq, 2 times 0.9 times 0.1 would give us the frequency of how often we would encounter a heterozygous organism. And finally, if we were to put 0.1 in for Q, 0.1 squared would be the percent chance that we would encounter a homozygous recessive individual, and if this is done correctly, it should add up to 100% of our population, you can simply try this with a calculator. So again, the Hardy-Weinberg equilibrium equation is very useful for population geneticists because it allows us to get a rough idea about how likely it is to encounter homozygous dominant, heterozygous, and homozygous recessive individuals within a population, again under the condition that the population is in genetic equilibrium. This concludes this video on calculating allele frequencies. Before we move on to discussing the factors that drive the process of evolution, you can take a look at the practice problems below that mainly involve using the Hardy-Weinberg equilibrium to calculate the probability of specific phenotypes being observed in the population. So try the questions below before progressing on to the next series of videos.